A very good morning to one and all. Dear ladies and gentlemen, welcome to ATAL FDP second session. It is my great privilege and greatly delighted to take an opportunity to introduce our chief guest, Professor Joseph Selvin, Professor, Department of Microbiology, Pondicherry University, Pondicherry. He has completed PhD in pathology from Central Marine Fisheries Research Institute and MS University, and completed his postdoctoral fellow from University College Cork, Ireland. Professor Joseph Sir published over 134 research articles in international journals, cumulative impact factor around 270. And he received US patent granted for pigment production, and one more patent is applied for approval. Sir has been published 8 books and 16 book chapters in reputed publishers. Professor Joseph completed more than 20 innovative research projects and funds obtained from various national and international funding agencies like DST Indo-Russia, DST Indo-Ireland, IFS Sweden, DPT, DST, CSIR, UGC, MOES and INSA. Sir has successfully guided 14 PhD research scholars and mentor for 3 postdoctoral fellows. Professor Joseph is academic editor for PLAS ONE and reviewer for many scientific research publishers. Professor Joseph has invited in over 50 international conferences to present his expertise. Professor Joseph has been awarded more than 20 national and international research and administrative awards in his profession, like Fellow of National Academy of Sciences, recipient of Jawaharlal Nehru Award by ICAR in 2003, Indian National Science Academy Young Scientist Medal in 2006, INSA Professor LSS Kumar Award in 2006, INSA Anil Kumar Bose Award 2009, DST Young Scientist Project in 2004, UNESCO TEMA Grant and NGP Award in GRC of Marine Microbes in 2006, DPT Young Investigator Project in 2008, Grant in Euro Biofilms in 2009, Indian National Science Academy Award in 2009, Bioprospecting Fellow in 2010, ICMR International Fellow 2018 at Natyong Technical University, Singapore. Professor Joseph has delivered keynote address in many national and international conferences, especially Asia Pacific Marine Biotechnology Conference in 2017, University of Hawaii, USA. Sir as the course director for National Workshop on Biotechnics, Melankara Catholic College, and DPT sponsored short-term training courses on fermentation and bioseparation methods. Sir is the organizer for Indo-UK conferences on shrimp aquaculture funded by Royal Society UK. And Professor Joseph Sir is the chairman, Board of Studies in Microbiology, Bharathidasan University, chairman, Board of Studies in Microbiology, Pondicherry University. Sir is the task force in Marine Biotechnology and Aquaculture, Department of Biotechnology, Empowered Task Force in Marine Natural Products, Department of Biotechnology, Ministry of Earth Sciences. Sir is the member of Task Force in Kerala State Council Biotechnology Task Force, member of Kerala Biotechnology Commission, member of Gujarat Marine Biotechnology Board, principal investigator of Indo-Mexican collaboration on shrimp disease management, and Sir is the coordinator of a national marine microbiome project and a project partner in EU 2020 Global Microbiology Supporting Project. It is a great honor and privilege for us, sir, to have you with us today. Now I request our respected chief guest to address the participants entitled on synthetic biology and drug engineering and cast the light of your knowledge upon us. Thank you once again, sir. The platform is yours.
Thank you all. Good morning. So we will directly go to the presentation. 1 minute, please. So my presentation is visible. Any one of the participants, please.
Yes, sir.
Yes, sir. It's visible,
Okay.
sir.
Okay. So if any disturbance in case of any connection issues, so maybe you can inform your coordinator. They may contact me over phone, so we can resolve it. So resolve it quickly. Okay. So maybe another 1.5 hours to hours approximately we will discuss about synthetic biology and drug engineering. Particularly today I will explain about what are the developments in synthetic biology and basics as well as the approaches in synthetic biology. Then later we as you have the first session on metagenomic approaches, so I will be giving you maybe 2-3 slides about the insight about how the metagenomic is now advanced as well as the integrated with synthetic biology approaches. So then we will be seeing something about the application of synthetic biology in the creation of microbial cell factories and synthesis of some of the molecules, bioactive molecules and medicines, and its applications in human genome editing, etc. That's the way I will finish it. Okay. So, synthetic biology and drug engineering. So, first we will see the application of, before we are going to the synthetic biology, you should understand that there are many people are now discussing about the human genome editing. See, now there is the controversy across the globe. Everybody, even the general public, very well knows that the SARS-CoV-2 or the virus responsible for COVID-19 might have engineered, not natural. So, such a situation, is it possible for engineering a virus? Can we develop a synthetic virus? All these things I will give you some insight from the synthetic biology. So, first you see, everybody knows about the human genome editing. So, it's a controversial subject that is happening. So, everybody think about whether the creation of a super baby, whether the gene editing methods can lead to a creation of super baby. Is it possible? I will give you some insight. Before that, we should know what is genome editing. But we will see what is genome editing. First, we all know that the human genome. So we have around 3 billion plus or 3 gigabytes pair of sequences. So the sequences we have, that sequence means nothing. So the 4 alphabet A, G, C, T. 1 minute, please. I will make a pointer. Yeah, these 4 alphabet adenine, thymine, cytosine, and guanine, AGCT repeatedly written. So you have to just repeatedly write AGCT haphazardly. That is known as sequence or the DNA sequence. Then you have a doubt if it is a haphazardly written AGCT, how it became a sequence, how it became a genetic information. Yes, it became a genetic information. All the life in the earth, entire life, even the bacteria, so humans, animals, plants, anything. If you say anything is life, the life is made up of these 4 alphabet, AGCT. So that is our playground. That is the reason we are now thinking about. That is one of the reason now we are conducting this this seminar or the conference through online mode. We are of COVID-19. COVID-19 is what is the reason of COVID-19? Why COVID-19 is the fast transmitting, highly mutating? What's the reason? The changes in the AGCT. So everything happening in that AGCT, if small change will lead to maybe a good or the bad outcome, whatever it may happen. Okay, before that, a human genome is made of, I told you, around 3.5, I can say approximately giga base pair. So you have to be very careful, giga base pair. See, usually in computer science also the same, they will be using the bytes, kilobytes, and gigabytes, etc. But in the case of in genomic science, we use base pair, kilo base pair, mega base pair, giga base pair. So small change. Okay. So the 3.5 gigabase where we have the sequence information, but the studies are the recent after the Human Genome Project, the publications evidence that the humans have almost 25,000 genes, and approximately 19,500 genes are real protein encoding genes. Then you have a question: what for this long genome? What's the use? You may consider the other parts like fingerprinting regions, like mini satellite, micro satellite, whatever may be, all repeats, all tandem repeats, fingerprint regions, etc., etc. Even collectively, 3% of the genome is known and known for the human survival. And 97% of the genome is with unknown function or not yet characterized. That 97% of the genome is nowadays known as junk DNA, junk, J-U-N-K, junk DNA. So, okay, now we have a question. 3% of the genome is known for the human survival. 97% of the genome is do not have any known function, and it is denoted as junk DNA. Can we create a human, or can we create a life just deleting the unwanted DNA or unwanted sequence? That is the question. So that question was with a scientist known as Craig Venter. So the Craig Venter was the pioneer in this field. He was having this question along with, you might have known that Hamilton Smith, a Nobel laureate. So they were working together to create a minimal genome. What is a minimal genome? So this minimal genome is the smallest genome which do not have the unwanted DNA. As I told you, a human is having 3.5 approximately gigabase pair sequences. Just 3% of the sequences are known for the human function. 97% is unknown or not required for the human survival and other functions. That is known as junk DNA. Now we have a question: can we delete the 97% of the gene which is not required? So by considering that hypothesis, this was first attempted in a bacterium, the bacterium known as Mycoplasma. You might have heard about it. Mycoplasma. What is Mycoplasma? I'll give you some insight after a few slides. So the minimal genome is the smallest genetic element sufficient to build, very, very important, build a modern type of free living cellular organism. So yes, a minimal genome is the genome which is very minimally required for a free living life of an organism. The organism will survive freely. There is no doubt in it. So such a creation is known as minimal genome. So there's actually this minimal genome is the beginning of the synthetic genome. The synthetic genome is the beginning of what we are hearing about the news, the lab-based virus, all these things. So, but this is one side. But the synthetic genomes play a big role in actually creation of biofactories. Biofactories means microbial cell factories. So once we create a life with a minimal genome, we know all the genes and building blocks. So be able to engineer, be able to modify, be able to create, be able to advance that organism quite easily when considered to a natural genome. Okay. Now we'll see what is required for construction of a minimal genome. So we should have the knowledge of existing genomes. While you start working on a minimal genome project, we should have the knowledge of existing genome definitely. Then we should have some knowledge about the compared to genome analysis, otherwise known as bioinformatics or dry lab techniques, and protein sequence similarities, and we should know the homology modeling, the evolutionary analysis. That is very, very important. So the first minimal genome, the first publication appeared in actually 2005. Actually, at the end of 2004, this publication was published by this publication was published at Craig Venter Institute. That time it is known as synthetic biology group. You can just imagine, everybody thought that synthetic biology is a very, very, very new field. No, synthetic biology has emerged even 2005 before. You can see synthetic biology group. So the synthetic biology group that time was led by Hamilton Smith, a Nobel laureate. So Hamilton Smith is the responsible, his team is the responsible for the creation of first minimal genome or first minimal bacterium. What is that? You can understand this story. So this was a publication in PNAS in 2006. I told you 2000, end of 2004. No, end of 2005, November 2005. So originally the publication is appeared in January 2006. Here, Mycoplasma genitalium is a smallest organism, or the bacterium do not have the cell wall. Okay, but the bacterium can grow in pure culture. So it has only minimal metabolic pathway and minimal genomic redundancy. Means we have a 3.5 gigabase per sequence. Most of our sequences are redundant, the same sequence repeated. But in this small organism or the small bacterium, it is this mycoplasma. You might have known that is usually known as in microbiology PPLOs, pneumonia-like organism. It is a potential pathogen. Some of the mycoplasma are responsible for brain fever. You might have heard about it. Pneumonia. Okay. So they applied a technique known as global transposon mutagenesis. Using this technique, they find 100 genes are not useful to this organism. Then what they did, they silenced the 100 genes. Okay, they find these non-essential protein encoding genes. So the non-essential protein coding genes, they silenced all the 100 genes. They silenced it or deleted from the genome. So they identified 382 of total of 482 genes are present in Mycoplasma genitalium. So 100 genes are non-essential, then 382 genes are essential for the survival of Mycoplasma genitalium. So simply they disrupted all the 100 genes from the genome. They created a minimal genome. That was the first publication in 2005 or 2006, I can say. So very interesting fact is, while they disrupted the unwanted or the non-essential protein coding genes, the Mycoplasma laboratorium growth was accelerated. The growth was very fast. So you can see some interesting or positive effect on the bacterium, disrupted the 100 unwanted genes. So what is Mycoplasma? The Mycoplasma, simply known as molecules, are generally known as Mycoplasma. It do not have cell wall. All the other bacteria have the cell wall, but it do not have cell wall. So this bacterium can evolve by massive genome reduction. If you reduce the genome, we can create another mycoplasma, another mycoplasma genome, and free living mycoplasma can be generated with considerable deletion of its part of its genome. It's obligate parasite and it's free living, and it's very, 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 very small chances of genomic redundancy. Okay, now the question is, I told you the 100 genes are non-essential. So what is its fate in recombination? Did you know we are all evolving through either homologous or heterologous recombination. So this genetic recombination is a very important role in the evolutionary aspects. So what is the fate of these 100 genes in the evolutionary as well as the recombination? Nobody knows that. People are still working on it. This is very, very important consideration. Then, after that, in 2008, the second publication appeared from the same team. It is also led by Hamilton Smith. That was the complete chemical synthesis. Their first time, they're completely chemical synthesis and creating the first Mycoplasma synthetic genome. So the complete synthetic genome was created in 2008. So now we have to understand this is a minimal genome and completely chemically synthesized genome or artificial genome. Now we should understand the architecture or the profile of the synthetic genome. So then we will know how to create a synthetic genome. What are the methods involved in? First, you should know this architecture. What is its architecture? See, I told you previously, they created the completely chemically synthesized synthetic genome. It has all the genes present in the wild type Mycoplasma genitalium, but only one gene was completely disrupted. That gene is known as MG408. The MG408 is a peptide methionine sulfide reductase, or simply I can say it is a virulence gene. So MG408 is responsible for pathogenicity. They deleted that virulence gene, they inserted an antibiotic marker in this region, MG408 region. Okay, now you'll see the design. So this design contains, the first level contains watermark. Okay, the second level and third level, they inserted lot of transverse zones. Transverse zones, you can say fingerprint region or mobile genetic elements. So these transverse zones are actually, you can see 2006 and 1999 means some of the transverse zones are discovered in 2006 from the Mycoplasma genitalium genome. 1999 means the transverse zones which are discovered that time from the Mycoplasma genitalium genome. Both already known transverse zones was listed, the second and third level. And the fourth level is very important, synthetic DNA cassette. Okay, what for this synthetic DNA cassette? I will explain after a few slides. Then everybody known that is protein coding genes. So that's now the 1, 2, 3, 4, the fifth level assembly is known as protein coding genes. Okay. Now see the fifth level only protein coding genes. The 4 levels are entirely different things are there. These things you cannot see commonly in natural genomes. Okay. And the 4th and the 6th level, everybody, it is quite common, structural RNAs. It is obviously present in all the natural genomes. So what is unique in this unnatural genome? 2 things are very, very unique. The first one is watermark. The second one is synthetic DNA cassettes. So we will be discussing about why we require a synthetic DNA cassette. What is the watermark? These things we will be discussing. See, if you search the literature, you just Google it, you will understand the Greek vendor did secret messages, encoded messages he kept in the watermark. So watermark, see, you have a doubt, watermark, transverse zone, synthetic DNA cassette, protein coding gene, structure, everything actually oligonucleotide. Okay, all these design is made up of simply AGCT. You don't forget it. Whatever the name, but everything is made up of AGCT repeats. Okay. Now, the watermark also oligonucleotide sequences. See, these oligonucleotide sequences carry secret messages. So, what the point I wanted to say is, almost 2008 to 10 is the beginning of synthetic life or synthetic bacteria on the earth. See, over a period of now many people are constructing synthetic bacteria. Now the general public also knows that what is a synthetic virus because of COVID-19. So now everybody aware of a engineered virus and its impact. So we never aware of a engineered bacteria, engineered animal, engineered plant, etc. So the time will come. Everybody knows that there will be a huge difference between a synthetic genome and a natural genome. How do you differentiate between a synthetic genome and a natural genome? With the COVID-19 research, I will explain you maybe after half an hour talk. You will be seeing, we will be entering that section where I will explain you that is known as reverse engineering. A technique known as reverse engineering. So reverse engineering was the technique used to create a synthetic virus. So here already demonstrated in 2010 a synthetic bacteria was completely chemically synthesized to oligonucleotide based bacteria. So there's this design, the watermarks are encoded for secret messages. What is the secret message? Simply Greek vendor. Greek vendor was encoded as these oligonucleotide sequences. Vendor Institute means Greek vendor institute. The name was encoded as these oligonucleotide sequences. Hamilton Smith is encoded as these oligonucleotide sequences. That's the way. Now you'll see, this is an indication over a period of time, the earth will have both synthetic life and natural life. So how do you identify the difference between the synthetic life and natural life? This sequence-based information only. So this sequence-based information, if effectively decoded, we will be understand who created, when it was created. All this information will be available. So that is the reason they given the watermark. The second, 5 to 7 kb cassette. That is actually, I told you around 383. See, so okay, not 380. This is a completely chemically synthesized genome. It has 583 kb. See, I told you this this is correct only. 383 genes are present in this genome. So the 383 genes you just imagine the 380 genes, the sequence information is available with us. Now it is chemically synthesized. Now you know the sequencing and analysis, you might know the word assembly annotation, genome assembly and genome annotation. Genome assembly means, we have no time, but I will tell you, as the genome sequencing are happening through the shotgun method. Shotgun method means the whole genome is spliced into pieces, millions of pieces or thousands of pieces. So the pieces are undergo sequencing. Then you get the only sequence information. Then you assemble all the pieces into one genome. Okay, that is assembly. After you are assembled it, then you have to identify the location of the genes, non-coding regions, fingerprint regions, etc., etc. That sort of marking is known as annotation. So assembly and annotation is an important technique. Now you'll see this assembly. I will tell you. See, you have 384 genes. The gene sequence information is available. It is already chemically synthesized. Now, how you will assemble it? We are going to create a real synthetic genome and real life. Okay, it is not virtual. It is actual. It is not happening in a bioinformatics tool alone. No, it is happening in the wet lab also. We are creating this clone and we are creating the synthetic genome and synthetic bacteria. So now you see all the genes you cannot, you can say A, B, C, D, like all the 383 genes, you just assemble it 1, 2, 3. Okay, very good. But see, a gene expression require, you cannot see one gene start expression, you might have known the finally start codon and stop codon downstream regions. After immediately another gene will start, means no gene will get expressed. The genes become not functional. So that's the reason the genes require the word gene loci. Gene loci is very, very important for gene expression. So the gene loci, it is a separation between the sequences. Okay. So the gene placed in gene present in one place and it is separated by naturally different sequences. So here it is a completely synthetic genome. There is no natural separation. So we have to provide a synthetic or artificial designs. That is, it is just like we are writing a sentence. You are writing a sentence, there should be a space between one sentence and second sentence, even one word and second word. So that space is known as cassettes. So the cassettes are nothing, the space between the genes. So the genes are arranged and organized. The space between the genes are known as cassettes. Okay, synthetic cassettes. So these cassettes are unique in synthetic genome only. You cannot find it in the natural genomes. So what is happened the next? Then thereafter, after 2008, the next publication claims chemical synthesis, not chemical synthesis alone, rewriting of a bacterial genome. So complete redesign of a bacterial genome to achieve design flexibility and enhance the biological functionality. So as of now, we have the synthetic Mycoplasma laboratory or synthetic bacteria. The version 3 is available now. So the version 3 carries 473 genes. So it is synthetic bacterial design and discovery is continuously going on. Okay. Now I will explain few minutes about what is synthetic biology. Actually, we should understand the basic concept of synthetic biology. That is very, very important. See, the term synthetic biology was defined, design, construction, and improved and novel biological systems. See, this synthetic biology that uses engineering principles. I can very simply explain what is synthetic biology this way. See, in general, the genetic engineering or the genomics originally start from cell biology. Am I correct? The cell biology transformed into genetics. You know, Mendelian genetics. We know that traditionally Mendelian genetics. So genetics transform into genetic engineering. Everybody knows that. Genetic engineering still today we are adopting the tool, genetic engineering. So genetic engineering transforming into cloning. So you might have heard about the 96 very important news, that is Dolly, a sheep cloning. So nowadays cloning is not in big place. The reason is genome engineering came in. When genome engineering came in, after cloning, genomics came in, Human Genome Project. That is the 2003 human genome, the first draft was released, and the field genomics become the frontier area of research. Everybody is working on genomics. So genomics then leads to genome engineering. What is the difference between genetic engineering and genome engineering? You should know that. So in genetic engineering, we select one gene and we synthesize or we do a PCR amplification and transfer the gene into a vector. So the vector is transformed into a host, maybe E. coli. So the E. coli will express the gene. So that is known as genetic engineering. Or the same vector we transmit into a plant, then it is known as transgenic plant, whatever may be. Okay, that is genetic engineering. But genome engineering means not single DNA, sorry, not single gene. It's multiple gene. A part of the genome can be deleted or can be changed. So it is just like tailoring. How we are doing the tailoring work, we just redesign the genome, we just delete some sequences, insert some sequences. So that usually that tool is known as CRISPR-Cas genome editing. Okay, so now the genome engineering came. So after the genome engineering, the field is now advancing into completely using the engineering principle, switch off mechanism. That process is known as synthetic biology. Okay, now you understand. This is the development in the biology. So in synthetic biology, a cell is treated like, a cell is actually just hypothetically considered as a computer. It is a computer analogy. Means simply a cell which is having a computer having hard drive and softwares. Very similarly, here also the software means you can say the DNA. The DNA is carrying the genetic information, act like a software. And the hardware means cytoplasm. The cytoplasm and the chromosome, and the DNA is present in the chromosome. The chromosome is the operating system. When the gene should express, when the gene should silence, everything, the information present in the chromosome. Then the cytoplasm acts as a hardware. So that analogy was people are now visualizing a cell as a computer. So we have to work with a cell how we are working with a computer. So that phenomenon is now basically known as synthetic biology. Or I can say simply say, you know, lot of genes. You have to program the genes. You have to redesign the genes. Now you synthesize the genes and create a partial synthetic genome and convert it to a cell. And the cell is known as cell factory. So initially it is a cell. Now the engineered cell is known as cellular factories or cell factories. Okay. So before, you should know few terminologies used in the synthetic biology. The important terminology is repressors. So the repressor means repressor plus oscillator. Repressor means switch off. You know, all the genes have downstream and upstream. Switch off, switch on. So all genes are working or genes are expressing based on the external stimuli. So one stimuli is essential for gene expression. Similarly, another stimuli is essential for the gene, the turn off the gene expression. So both are happening naturally also. But in synthetic biology platform, we are using the molecular switches known as repressors. So the repressors are engineered genetic circuit designed to control the gene expression. Repressor means inhibition, oscillator means turn on or induce the gene expression. Okay. So good example is, you know, this squid and this Lux gene. Lux gene is otherwise known as the fluorescent gene. So the fluorescent gene expression can be controlled externally. See, I will show you one of our laboratory experiments. This is, we did a laboratory level. We had published this already. So see, this is a bioluminescence in a ordinary test tube. So this bioluminescence expression of a cell, a bacterial cell, can be controlled with heavy metals. See, different heavy metals. This is zinc, copper, mercury. You can see the differential expression pattern. The expression is widely varied between the three different heavy metals we tested. So what the point I wanted to say is the gene expression and the gene switch off, switch on, gene expression and gene inhibition have to be operated, have to be controlled externally. Then it is just like a switch off, switch on mechanism. So the external switch off mechanism is known as repressors. Okay. See, synthetic biology is a interdisciplinary field. So it works between biology, engineering sciences, computer sciences, and most obviously bioinformatics. So as I told you, the genomics and further it is evolving into functional genomics and functional genomics into protein engineering. That protein engineering evolved into chemical biology into metabolic engineering. Now you might have heard about the systems biology, very important field in bioinformatics. The system biology, then it is evolved into bioinformatics. Now you'll see the actually this is one way, but actually the synthetic biology platforms are evolving from bioinformatics. I can put in reverse order. So the bioinformatics, I think 90s to 2000 we have the field of bioinformatics, and 2000 2005 we have the genomics and proteomics, and after proteomics the proteome means metabolome. You might have known that most of the metabolic pathway is actually led by the pool of proteomes. So the functional proteomes are known as metabolomes or metabolomics. Then the proteomics evolved in metabolomics. So from the metabolomics, the complex network, the metabolic network analysis. See, we have the glycolysis, HMB shunt pathway, or Krebs cycle, whatever may be. So each cycle have its own metabolic pathway, and all the metabolic pathways are interlinked. Such a complex network analysis is very difficult. Then, after metabolomics, the field evolved into systems biology. So the system biology then evolved into synthetic biology. So synthetic biology is a recent emergence, but as I told you, synthetic biology was OBBS adopted in 2005 by Hamilton Smith and Greg Winter team. Okay. See, now we have to revive the central dogma. You know the central dogma, gene to mRNA or gene, RNA, and protein. So that simple concept we can explain very elaborately as gene to mRNA, mRNA into proteins, into metabolites, because all the proteins become metabolome or metabolites. Now, the metabolic pathway stimulation, when the pathway should act, when the pathway will act, so when the pathway becomes functional, that is known as stimulation. So similarly, each field, the pool of genes and analysis is known as genomics, the pool of RNA or the transcript analysis is known as transcriptomics, the pool of protein and its analysis is known as proteomics, the pool of metabolic pathway and its analysis is known as metabolomics, and the stimulation-based metabolomic pathway analysis is known as fluxomics. What is fluxomics? I will explain to you after a few slides. Fluxomics means like a balance mechanism, reactant, product. You know, if you take a chemical reaction, all the reactant and products are balanced. That is known as the chemical equation balancing. You might have heard about it or might have known that. So that sort of flex balance also very important in the metabolomic analysis. That sort of analysis is known as fluxomics. Okay, see, now we will see the synthetic biology have a lot of applications, all these things. First, I will explain two softwares. If you are interested to work with synthetic biology, there are many interesting freely available softwares are available. That is known as CAD. CAD means computer aided design softwares. So because synthetic biology is mostly it is a design tool. We have to create a life. First we have to design synthetic DNA. So this GenoCAD is a freely available software. If you are interested, you can start working today. You become a synthetic biologist, definitely. It is a beginner. So it is freely because you can learn your own. You can learn your own, not required anybody's help. Because the tutorial, everything is available in the GenoCAD software. So because it is a web-based tool, you need not install any Linux or any other platform, not required. So this is a web-based tool to design plasmids, artificial gene networks, synthetic genetic systems. So we have to create different genes and its parts. Okay, so that can be created here. Then you can design. Finally, you can stimulate the metabolic pathway also. See the features of the GenoCAD. It is a database of high quality genetic parts. It is a web-based application, no installation required. So it is quite easy, and easily we can work with GenoCard. So you can import different genes, different enzymes, different metabolic pathways. Then you can create a new synthetic genome or synthetic metabolic pathway, and you can check it. You can analyze, simulate it whether it will functional or not. The another one is Tinker Cell. Tinker Cell is another CAD for synthetic biology. So this software also, for example, engineered basal line. So you have the basal line genome and metabolic pathway. You can change that metabolic pathway with the different. I will be explaining you really what is a metabolic engineering after a few slides, where you will understand very clearly. So you can insert the different metabolic pathways, and you can test, and you can analyze, and you can create new metabolic pathway for your new synthetic genome. Okay, now we will see real approaches. So far we just seen about the bioinformatics tools and the basics. Now we will see the first one I will be explaining you is real application based approaches. Creation of microbial cell factories. So how to create a microbial cell factory, or what is metabolic engineering? I will be explaining here. See, so 1 minute, I will be giving you 2 minutes break. You can just be 1 minute. It should be online, just for 2 minutes break. So thank you for all. We will start again. 1 minute. Okay. See, genome-guided microbial cell factories. You know what is a cell factory? Before that, I will explain you the chemical process, the bioprocess. The chemical process means all the chemical industries. Nowadays, we know the chemical industries are mostly discouraged because of the green energy and green technologies. Otherwise, I can say you the green chemistry. The green chemistry means zero-based production process are known as green chemistry-based industries. So all the chemical process actually leads to industrial pollution. Everybody knows that. But the bioprocess or the fermentation based process are less pollution producing industries, But again, the problem is, again, we wanted to reduce the pollution. So we have to apply the two techniques. One is metabolic engineering, another one is genome engineering. Both are actually just, I can say, the two sides of a coin. Okay, both are good together, metabolic engineering and genome engineering. So that principles are applied in bioprocess. So thereby the bioprocess become zero waste production process. That is known as greener bioprocesses. So one side, zero waste production process, another side, the production capacity was many times, multifold increased, many times increased. The production capacity is increased, and the pollution also, there is no pollution or zero pollution or very, very less pollution. So that sort of industries was proposed and people are working on it. It was achieved, I can say. Many industries already they are working on it. So, just 1 minute. Is audible? You are hearing me?
Yes, sir.
Thank you. So, see, now you will see, I am saying bacteria as a chemical factory. So you can see the bacteria is a single cellular organism, okay? I am saying the cell as the factory, chemical factory, okay? So what we are providing is the starting material. So we should provide only carbon. Carbon means, I can say, how I can say, in the case of the carbon means we have to provide only carbon as the substrate or the nutritional source. For example, paddy straw or any agro-industrial waste as the as the nutritional source or carbon source. So the cell have to create, using its own metabolic pathway, the cell have to produce all the products. For example, antibiotics, vitamins, enzymes, hormones, vaccines, everything, the cell has to produce only from the carbon. So we provide only carbon as the source material. So then this process is highly economic, highly efficient, and greener process. There will be no waste. Okay. Now see, this type of bioprocess aiming to produce high value chemicals. They wanted to replace all the existing chemical processes. Example, hydrogen means fuels, next generation biofuels, methane, methanol, ethanol, and engineered products, rubber. See, it is almost achieved. Did you know, most of you know that those who are from Kerala or South India might know very well the rubber plantation and its uses. Though the rubber is useful, but rubber also causing lot of ecosystem damages. Then the rubber plantation can be completely replaced. Rubber plantation is actually not required once synthetic rubber, the bioprocess was developed. Almost achieved actually. So the production of rubber, plastics, that is called bioplastics, the synthetic fabrics, and synthetic fabric means the silkworm, you know that any naturally produced natural bio or any metabolic pathway based product have to be produced in a bacterial cell, even in a plant product or animal product, whatever it may be. And bulk chemicals, vitamins, therapeutic agents, even butanol. Yes, almost it is planning to replace the fossil fuels. So the butanol as the jet fuel, so that the white petroleum can be replaced. So that's the way. These are all the requirement. How to create a cell factory for each of these products. See, what is the limitation? You might have a question. Already bioprocess technologies exist, genetic engineering is available, mutation methods are available. All these methods that is known as strain improvement methods. All these methods are now not sufficient to develop a robust, or I can say hyper producing strains, and the economic industrial production pathways are not achieved. For example, you might have known that bioplastic, polyhydroxyalkanoates or polyhydroxybutyrates. Why it is not existing every part? Not it is exist for human use. Not it is exist for all the places as a local market. Not available. The reason is economy. The production economy is very high. How to reduce the production economy? That is redesigning of a bacteria. So what is redesigning? So you see, this bacteria, if you provide carbon, it is metabolic pathway. It is metabolic pathway always synthesizes metabolic, the end products For example, metabolic end product may be an acid, maybe an enzyme, whatever may be, or intermediate metabolic products. That products are maybe useful products. By the same time, the lot of metabolic pathway products are, I can say, it is not useful for us. So that products are, I can say, waste. Again, waste accumulation is there. So we have to redirect the metabolism to a specific product. For example, if you are aiming for a rubber, then the metabolic pathway should be redesigned to synthesize only rubber, no other metabolic product. And the bacteria should not have any storage product, no excretory product, no defense product. Everything is deleted. The genome is completely redesigned to synthesize and redirected towards the synthesis of a particular product. So that field is known as metabolic engineering. What is metabolic engineering? See this simple example, I will explain you. This is a genetic pathway or metabolic pathway. See here, gene 1, gene 2, gene 3 is required. This is A. A is, as I told you, carbon, like substrate. We provided carbon. Carbon is A. So the A is converted into metabolic intermediate product B. B is converted into metabolic intermediate C. C is converted to D. Okay. So a bacteria can produce D, maybe an enzyme. But we require synthetic rubber. So this D is the substrate for the production of rubber. Can you imagine? So now the bacteria do not have the metabolic pathway to produce a rubber. Then how we can redesign the bacteria to start synthesize a rubber? How? We have to redesign. Now you can see we are deleting the gene 3 first. So you do not have enzyme 3. Okay, there is no product D. Then we are inserting a new gene 3 and you have a new enzyme 3. So that leads to a next metabolic intermediate product D2. Okay, not D, not as a metabolic end product. So this is again a metabolic intermediate product. Now I am inserting another gene, that is gene 4, required for an enzyme. That enzyme convert the metabolic end product D2 into what I am expecting, maybe a rubber product E. You understand me? This is known as metabolic engineering. All of you got it? Okay. Now, there are many metabolic engineering based synthesis is happening. Example, metabolic pathway of meconic acid. Meconic acid production in Corynebacterium glutamicum. So another one is, okay, before another example, but the metabolic engineering sometimes end in actually mishaps. Mishaps means failures. See, one such example is, see, this is a methanol. Okay, methyl alcohol. So the methyl alcohol is always converted into alcohol dehydrogenase enzyme, converted into formaldehyde. You might have known that, formalin. This is one of the reasons you might have known the illegal what can I say, the illegal alcohol. The people may drink illegal alcohol. They immediately you can heard about the news, they die immediately. The reason is the body converts. Yes, what is the illegal alcohol contains? The illegal alcohol contains methanol. Methanol is otherwise known as wood naphtha, not ethanol. Ethanol means ethanol is converted to acetaldehyde. It is not harmful to our health. I cannot say not harmful. Comparatively, it's not harmful. But in case of methanol, that's methyl alcohol, it is converted into formaldehyde or formalin, immediately fix our body. That's one of the reasons that those who are drinks illegal alcohol, their eyesight, they lost the eyesight immediately. Okay, it's okay, leave it. Now, we are engineering a metabolic pathway for the production of polyhydroxyalkanate, bioplastic. Okay. So, this pathway is happening in a methylobacterium bacteria. Here, this methanol is converted into formaldehyde, and formaldehyde is for taking two pathways. Either one pathway leads to carbon dioxide, that is a metabolic end product, or another pathway leads to polyhydroxyalkanates. But ultimately, we know that if we delete this pathway, the entire metabolic intermediate, that is formaldehyde, will convert into polyhydroxyalkanate. That's a good approach. Thereby, we can delete the unwanted pathway. We redirect the entire pathway towards the synthesis of polyhydroxyalkanate. But one problem is here. See, the metabolic intermediate accumulation here, if it is converted into two ways. The metabolic intermediate accumulation means the formaldehyde accumulation is very less, immediately is converted into both the ways. But if you delete this pathway, what will happen? The intermediate will sustain for some time because it converted into the PHA pathway may take some time. The time limit, that is known as fluxomics, the flux balance analysis. Within this time, what will happen? The cell will die because of the accumulation of formaldehyde. Okay. So the metabolic networks are actually, all the metabolic pathways are very complex. Such sort of mathematical modeling to predict and to redirect a metabolic pathway to a synthesis is known as synthesis. We need a lot of mathematic models, otherwise known as algorithms. Such algorithm-based analysis and metabolic pathway or metabolic network analysis is known as systems biology. So system biology knowledge is basically required for the synthetic biology work. Very, very important. Okay. This is flux balance analysis. I told you, no, you are providing carbon as the substrate, and carbon is converted into different metabolic intermediates and final products. But see, this metabolic intermediate balance should be maintained appropriately. Otherwise, what will happen? Some metabolic intermediate will kill the cell, or the metabolic end product, accumulation of a particular metabolic end product will kill the cell. So to avoid that, you have to calculate the flux balance analysis. That sort of calculation and analysis is known as fluxomics. You understand me, no? Okay. So now we will see one real example, the synthesis or metabolic engineering and production of artemisinin. So the artemisinin is an anti-malarial drug which was awarded Nobel Prize in 2015. Everybody knows that. So the artemisinin actually synthesized by a plant, a Chinese plant, Artemisinia annua. See, synthesis and production of this plant belongs to China. Then every country, they have to pay to China for patent or all these things. Instead, if you have a technology, produce the same artemisinin in a bacterial cell, anybody can produce it. So is it possible? Synthesis and production of artemisinin in bacterial cell is possible because it is a plant product. It is a plant-based, secondary metabolites. So that can be produced in a bacterial cell is possible or not? It is possible. How? We will see. See, this is a symbol. Okay, I will explain with the next pathway. See, here also little confusion. Okay, here I will take. So this is the real metabolic pathway happening in a plant cell. Okay, here carbon dioxide and sunlight. So the carbon dioxide sunlight converts. You know that sugar. Am I correct? The carbon dioxide is converted to sugar. So the first sugar. The sugar is converted into acetyl-CoA. Everybody, even all the life in the earth is having this metabolic intermediate product. That is our playground, acetyl-CoA. So acetyl-CoA is in Krebs cycle or Krebs cycle. So we did not bother about that cycle. So we need this metabolic intermediate and how to redirect acetyl-CoA into synthesis of artemisinin. See, first we understand acetyl-CoA is the metabolic intermediate product after sugar, after glycolysis, all these things. Then it entered into acetyl-CoA, TCA cycle. From the TCA cycle, it is converted into FDP, farnesyl pyrophosphate, farnesyl diphosphate or farnesyl pyrophosphate, FDP, farnesyl pyrophosphate. That is correct. So farnesyl pyrophosphate is converted into amorphodiene. Amorphodiene is converted into artemisinic acid. This is the pathway. Now you will see A bacteria is having acetyl-CoA, but bacteria do not have this pathway. Farnesyl pyrophosphate amorphodiene pathway. A bacteria do not have. If we are able to provide this pathway in a bacterial cell, a bacteria is able to synthesize artemisinin. Okay. Now you will see. So synthesis of a artemisinin E. coli. So what to do? So this farnesyl pyrophosphate and other metabolic intermediate product IPP. See, for example, inosine pyrophosphate or the DMAPP, dimethylallyl pyrophosphate. So all these metabolic, these three things, IPP, DMAPP, farnesyl pyrophosphate. These three metabolic intermediate genes responsible for artemisinin synthesis was inserted. So this is TCA cycle. So the TCA cycle, first it starts with acetyl-CoA. The acetyl-CoA, part of acetyl-CoA cycle, not going through the TCA, instead it redirected to another pathway, that is IPP-FEV pathway. So the IPP-FEV pathway was a engineered pathway, newly inserted pathway in the bacterial cell. So now the bacteria start to synthesize artemisinin. So artemisinin, all the plant-based products can be synthesized in a bacterial cell. It is possible. Okay. So what is the problem in actually while you are performing a genome engineering and expression of a product in a bacterial cell? Actually, the artemisinin is toxic to the bacterial cell. You might have known that. Again, flux balance analysis is very important. How much concentration of artemisinin the bacteria can survive? So that have to be calculated. So based on that analysis, we have to create this design. Particularly, we have to provide reproducible promoter control. See, the promoter should be externally controlled. So that external control switches are known as, did you just remember, represslators. So each gene should have a repressolator. So see, you can just come back to the Great Vendor or the Hamilton Smith design minimal genome, where they provide 5 to 7 cassettes. Cassettes are separating sequences between the genes. Here I am saying the genes responsible for IPP, DMAPP, FEP. So all these metabolic pathway genes you have, very good, but the genes should be separated by different promoter controls. So that promoter controls are known as represslators. Thereby, you can induce the gene expression consecutively, one by one, so that we will get first metabolic intermediate that is converted into DMAP, then another metabolic intermediate converted to FEP. FEP is further another enzyme convert the FEP into artemisinin. That sequence is very, very important. If the sequence changes also, there will be no expression. So that flux balance analysis taken care of. That flux balance analysis is known as fluxomics. So usually you might have the first talk, metagenomics. So usually we construct the metagenomic libraries with high copy number plasmid. Yes, the high copy number plasmid, high number of plasmid are the sequences. Sorry, the heterologous gene will be present in high number and high copy number plasmid. There is the number of copies of the gene will be increases with increase the production of our enzyme in a metagenomic library. But such sort of metabolic engineering or the toxic metabolite based engineering process, you should use low copy number plasmid. That is best. Otherwise, what will happen? The high copy number plasmid, the metabolic intermediates will be, the expression of metabolic intermediate and intermediate product accumulation will be very high. The cell will die. Okay. Okay. So now, what's the point I wanted to summarize is before going to the next application. See, what is the difference between genetic engineering and metabolic engineering? What is the difference? Here, we express now multiple genes. Genetic engineering means expression of a single gene. That is the basic difference between genetic engineering and metabolic engineering. Okay. So now another technique, genome editing, creation of bacterial robots. bacterial robots. That is another cell factories using CRISPR-Cas gene editing tools. So I'm not giving much information here. I will go into the next session, that is creation of a or gene editing in humans. So gene editing in humans, we thought that it is not possible, but it was possible. It was already done. It was already demonstrated, but unethically, not ethically. There was a Chinese scientist in 2018. At the end of 2018, there was many newspapers have this news, Lulu Nana controversy. So He Jiangqi, the scientist is known as He Jiangqi. The He Jiangqi shocked the entire scientific community. He claimed that the first gene-edited babies have born. The first gene-edited babies are named as Lulu and Nana. Who are all Lulu and Nana? Actually, Lulu and Nana is the nicknames. But in Google, if you put Lulu Nana, you will get many information, many news, and many updates about these two babies. What happened to these two babies? Actually, these two babies born to a HIV positive parent. So usually, there was a chance the HIV positive parent can give birth a baby may carry HIV positive. There is a chance. So considering that fact, He Jiangqi approached the parents of Lulu and Nana. He explains that the babies, the gene responsible for the HIV virus infection or the recognition in the white blood cell, a gene is known as CCRB5 protein, CCRB5 protein, chemokine receptor protein. So the CCRB5 protein is deleted, so the HIV virus cannot be able to recognize the white blood cell. So white blood cell it cannot recognize. So what he claimed, he informed the parents that while at the time of in vitro fertilization, the gene responsible for the HIV infection is CCRB5 protein or CCRB5 gene can be deleted from the babies. If you delete the CCRB5 gene, a HIV virus cannot infect a person. So considering that hypothesis, he taken the consent of the parents. He deleted the gene from the two babies' genome at the time of in vitro fertilization. Consequently, the two babies born, and he sequenced the baby's genome, and he very successfully the baby do not carry twin babies do not carry the gene responsible for HIV infection CCRB5. That's very successful. And the entire scientific community was shocked because it is unethical. Human genome editing is not permitted in any of the countries. We should not perform a human genome editing. It was the first human genome editing was happened. So that was the twin girls born to prevent them contracting HIV. But there is no publication about how this gene editing happens. But what went and what wrong, how it is unethical thing happened, there are many publications. That is known as CRISPR baby scandal. You can find many publications explains about the wrong things, gene edited babies, what went wrong, what could go wrong. So one point I wanted to explain, the He Jiangqi is now in jail. He sentenced to jail and all his research was banned, but the two babies are now growing actually. So the scientific community is actually observing these two babies. So the CCRB5 protein, originally he thought that it is responsible for HIV infection. Really it is responsible for HIV infection, that is true. But the CCRB5 protein have another role also in the humans. So if you delete the CCRB5 protein, what will happen? I will explain after a few slides. So in between, actually another scientist, because of the synthetic biology development, so the challenging death, the new project of Greg Winter, what he says, 2045, death became optional. The scientist named Cardairo from Massachusetts Institute of Technology, MIT, Massachusetts Institute of Technology, USA, he claims that the death became optional. How? So that was the claim. After that, there was a real project also. So the project, that is current project going on with Greg Winter, no much publications, but hope we will have many publications soon. So that is how to cheat death. See, we know that we have a birth date, we have a death date, every human or every life. But the great vendor claims, I can say it is the concept little actually their over expression, cheat death. That is not correct. This is a concept of longevity. How to expand, how to extend the lifespan of a human. So that project is known as, actually the project is known as longevity project, human longevity project. How to extend the lifespan of a human. How to extend the lifespan of a human. We will see a few things that also happening through genome editing. See, frequent did an analysis. The analysis he find that every human, yeah, for example, a small percentage of the people, did you know actually, yeah, the people who are surviving 100 years, that is mean centurions, in the world, 5 zones or 5 cities or the 5 regions, the people are surviving 100 years. Their normal lifespan is 100 years. That zones are known as blue zones. Example, Loma Linda, Igeria in Japan. So you can just Google it. You will find blue zones of the earth. The 5 zones are places where people are always, means normally the people have 100 years of life. So the analysis are going on. So now I am speaking about in general, the people reach 90s are very difficult. Okay, see, everybody now knows that. While we are crossing 40s, in India, we have the greatest burden of hypertension and diabetics. 40 to 50, we are under the time of diabetics and hypertension. Then every time we are running the days with tablets or what can I say, medications. But what the people reaching 90s are very rare, but it is some people are reaching 90s and 100 also. But Greg Vendor what says, Greg Vendor is from this project, Greg Vendor is hoping that we can make a normal lifespan of a human is 3 digits. At least the human have the lifespan of 100 years. Is it possible? So you see this project about the what sort of genome editing they have planned. So for example, one of the case study. See, the company, the Great Vendor company, detected cancer in 5% of the people over the age of 50. But they did not know that they carry the cancer. And 40% of males, those who reach 50 years of their age, they won't cross to 74. Very similarly, 28% of the women who reaches the age of 50, they won't make it to 74. And when here, one interesting analysis from this study is, compared to the males, the women have the longevity by nature. By naturally, women have the long lifespan when compared to the males. So obviously, 40% of the males and 25% of the females have short lifespan. They cannot make it to 74. What Greg Vendor is claiming, by making genome analysis of all the humans, we can predict and we can prevent cancer-causing genes, or we can delete cancer-causing genes, as well as diabetic genes, hypertension. There are genes responsible for hypertension also. All disease condition, physiological stress, the genes are responsible. So that genes can be deleted, thereby the human will have at least 3-digit lifespan, minimum 100 years of lifespan. So one of the, they have actually a lot of examples. One of their analysis with the 18-year-old patients, means girl, they find several oncogenes on their genome. So this human longevity project, frequent human longevity project, predicts that. So now they have identified this girl is having a lot of oncogenic genes. So once this girl grown up, she almost reaching the age of become a cancer patient. Means the oncogenes become expressed, undergoes the oncogenesis pathway, and she developed with tumors and cancer cells, etc., etc. While she reaching at that stage, the Great Vendor team will have the remedy for it. So that is the way this project is going on. You understand, we know? The next interesting project going on is designer genomes and beginning of synthetic life, or designer baby project. So people are working on this line. because why this area is growing because of Lulu Nana. So I told you Lulu Nana do not have the CCRB5 protein responsible for the HIV infection. But now this baby is very recently analyzed by a team. They understand that these two baby will have super brain or super memory possibility. So what's the reason is the CCRB5 protein, if you delete, they will have high cognitive ability. Cognitive means what can I say? The memory as well as the IQ ability. So the two babies' brain will develop quite fast. The babies might have superhuman behavior, but it may take time. Okay, I told you creating designer genomes and designer babies. Did you see previously the in vitro fertilization was a very rare technique, but now IVF is a very common technique. Previously, a baby embryo, killing a baby embryo was not allowed. Yes, even nowadays also, yes, it is unethical. But now we have embryo biopsy at the laboratory level. So that the laboratory level, through in vitro fertilization, the biopsy, we can be able to sequence it, we can analyze it. The baby embryo is having the required level of genetic makeup or not. If the genetic makeup is not up to the mark, what we expected, that embryo can be destroyed at the laboratory level, not at the mother's womb. So mother's womb it is unethical, but laboratory it is not saying as unethical, but it is a cell only. So that's the way it is. It was the technology was developed one side. In another side, see, the mother's womb was required. A foster mother is required for transmitting this in vitro fertilization, means the embryo, so that the mother may give birth a baby. Can we replace the mother? That also now the discoveries are going on. I don't know whether you're able to see this video, 1 minute video. I'll check 1 minute. Yeah, I don't know sound. You can just see it. 1 minute. I'll check the sound. 1 minute, please. Yeah, this is the maximum. Okay. Yeah, you can listen it. So what it says is actually artificial womb. So that was already developed and it is now going on. The research is going on. So IVF and artificial womb technology is available, then the creation of a baby in the laboratory is possible in the near future. Okay, now before going to the next application, maybe for another 10 minutes discussion, in between I wanted to explain, because you have a first lecture about metagenomics. So I wanted to explain the application of synthetic biology in metagenomics. See, 1 minute. So the metagenomics, how you might have learned very many things, sequence-based and functional metagenomics. I will explain about the functional metagenomics. This is one of our research previously. So creation of a metagenomic library. So see, we need a high molecular weight DNA. This is a pulse field gel electrophoresis. This is a normal gel electrophoresis. From the pulse field gel electrophoresis, be able to identify high molecular weight DNA. That is our requirement. We have this is the creation of a phosphate library. So the phosphate library requires 5 kb insert DNA. So that is high molecular weight DNA. So 40 kb insert. I am mistaken. 40 kb insert DNA. So the 40 kb insert was created. Then it was electroeluted. 1 minute, please. Yeah. It was electroeluted and it was cloned into a copy controlled thesis1 vector, a vector system. The phosphate library was created. This is a phosphate library. All clones. Library means clones. So plenty of clones, lakhs of clones together is known as library. This is a metagenomic library. But what is the problem in metagenomic library construction? So this is the robotic systems we usually adopted. 2010, 5 years we used this system for screening of metagenomic libraries. So the metagenomic libraries were screened for a lot of enzymes, bioactive drug leads, etc. We identified lipase from the metagenomic library and given a protein antibiotic from a soil metagenomic library. Everything is interesting, very good. But what is the problem? See, gene expression. So in a metagenomic library, actually the genes are present in a heterologous host. For example, originally the gene may be from a actinobacteria. So we create a library in E. coli. The E. coli cannot be able to support the gene expression because the metabolic pathway of E. coli is different from the actinobacteria. So the metabolic pathway of E. coli is different from the Pseudomonas. All these issues, so the metagenomic library screening is mostly failure. Mostly you will not find a hit. So for that, very recently, 2018, another new interface between synthetic biology and functional metagenomics. See, being able to get a expression of the gene is possible. This is a metagenomic DNA. So the cloning site, along with the cloning site, we have to provide a genetic circuit. I told in a repress later, gene expression, gene expression, switch on the gene expression, switch off the gene expression, repress later. We provide such a genetic circuit, what will happen? We can externally induce the gene expression. So any heterologous genes can be expressed. So maybe most of you have students and PhD and young faculty and scientists. You can definitely start your research on this line. This will become a very promising, very interesting, one of the new approach. But I have not seen not much publication. It was proposed. People are working on it. Okay. Now you'll see the synthetic biology application in metagenomics. See, they're realizing the potential of synthetic metagenomics. You know, there are many projects. One is the Human Microbiome Project. Another is the Great Wonder Global Ocean Expedition Project. I will be explaining something about Mammoth Genome Project. So what is happening with the Mammoth Genome Project? Mammoth Genome, you know, Mammoth is a Ice Age. You might have seen in the Ice Age movies. It is African elephant, which is not exist today. Can we bring back the Mammoth again? Is it possible? Yes. Now the things are going on that line. So the mammoth genome was discovered, means the DNA was found from the archaeological site. Yes, it is surviving in actually what can I say in a very cold era. It is surviving on the ice, so it able to the archaeological samples are freezed already naturally. So they able to recover the DNA. So they were start working on revive and restore. That project is known as revive and restore genome. Mammoth. So mammoth genome was restored. They wanted to bring back the mammoth. Is it possible? So already mammoth blood cells was now available in the laboratory. Okay, very interestingly, mammoth hair cells have been already grown. Mammoth fat cells already grown. So the laboratory level, all these pluripotent cells have been grown. So they achieved the cellular growth of the mammoth in the earth. Now they have to bring back the animal again, but that require the whole genome construct. That was the difficult for a long time. They were thinking of how to sequence and develop, how to get the whole genome of a mammoth. See, last February 2021, the first publication appeared in Nature. The million-year-old sequence, means genome of mammoth was the first sequence, the first level of sequence information was available. So from this sequence information, we can expect any time they can bring back the mammoth to the earth. Okay. Okay, I took long time, 1 hour plus. I thought of 1 hour. It's okay. I will finish my talk maybe another 5 to 10 minutes of time. Okay. See, uh, this is the one. This is one of the development. Another development you see, we are now undergoing the COVID situation. Rapid construction of SARS-CoV using synthetic genome platform. So you have seen a lot of insight about synthetic genomes. So synthetic genome is very important. So we be able to reconstruct a genome quite fast. See, nowadays the COVID is evolving very fast, rapid evolution. So such a rapid evolution, how to predict? Yes, no publication evidence says that this way this virus will evolve. No, we are unable to predict. But that sort of prediction is possible if you have a synthetic genome of SARS-CoV. That also achieved. Now, the rapid reconstruction of synthetic genomes of SARS-CoV is already available. But you have to remember one point: SARS-CoV itself is a synthetic virus. It's not a natural virus. But maybe things will happen. 2 publications are available to support engineered virus or lab innovated virus. It will take time. But now this platform, the synthetic biology platform, will help us to discover new drugs, new vaccines. So all these things are now possible. There is a hope. This publication appeared in 4th May 2020. So thereafter, many research is going on. Thereafter, the vaccines are coming out. So, okay. The next one, I speak much about synthetic biology and synthetic genomes. See, I start my talk with all life on the earth is made up of AGCT, adenine, diamine, cytosine, and guanine. Now I wanted to ask a question, whether we are able to create a life other than AGCT or extended alphabet or unnatural nucleotide based life is possible. So if you create a life using unnatural nucleotide, what will happen? So that reason you should know that hypothetical theories, I cannot say it is true. These are all theories about the people are speaking about alien life. But I wanted to explain who is alien. Who is alien on the earth? Even see at the end of the Trump time, there is a news. Everybody knows that India today. The news says that Donald Trump knows that aliens exist. Says former Israeli space chief. I don't know whether it is exist or not. That is not my aim. My aim is I try to explain you who is alien. Okay. For that, you should know 3 short, small theories. You have to just remember the theories. The one is Darwin theory of evolution. Everybody knows that. The second theory, you might have forgot about it. Stephen Hawking. Very recently, Darwin is the historical scientist. But our time, we have another theoretical scientist. Darwin also a theoretical scientist. So our time, we have a very famous theoretical scientist who is Stephen Hawking. So Stephen Hawking theory mostly support aliens. So Stephen Hawking clearly says that aliens exist. But again, all the participants underline the word theoretical scientist. These are all theoretical science. There is no experimental proof. The third one is hollow genome theory of coexistence. These 3 theories, if you understand, then you can clearly predict and clearly say that who is alien. This is about, I'm not speaking much about it. Darwin's theory, there is a biological evolution, chemical evolution, everything. So Darwin finally says that this is very important. It is not the strongest of the species that survive, nor the most intelligent, but the one responsive to change. So if the species responds to the environmental change only will survive. You understand me? So that was the theory of Darwin's theory of evolution. But our time, we have I told you 3 important projects. One is NIH funded human microbiome project. Another one is mammoth reconstruction project that I explained. The third project is Great Wonder Global Ocean Expedition project. So here the NIH funded microbiome project. So what microbiome project really working on? It is very interesting. Actually, all the humans we made up of 10 power 10 cells, but our stomach is having 10 power 14 cells. That is microbial cells. So now we previously we thought that humans are evolved as a human. But we now thought now it is clear that the humans are not evolved as human. Humans are evolving with their symbiotic bacteria. So a human is not alone. Human is always associated with her symbiotic bacteria. So the symbiotic genome and human genome together is known as hologenome. So the evolution is not alone. The not bacterial evolution, not human evolution. The evolution is hologenome evolution. So now the theory of hologenome evolution. You should understand it, but it will take time. We will discuss some other time about hologenome theories and hologenome evolution. The next theory, Stephen Hawking theory of aliens. Stephen Hawking says, the advanced aliens could conquer and colonize our planets. So that would be the end of our Earth. So once the aliens start communication with the Earth, so then the aliens will conquer, the Earth will end. There will be no human survival on the Earth. That is the Stephen Hawking theory. But see, here I don't want to explain. So I am not supporting this theory, none of the theories, but I am supporting only one theory, that is hologenome theory. Yes, hologenome theory is fact. We have a lot of experimental evidences are there. But another time I'll explain about the hologenome theory. But I wanted to say, who is alien? See, in 2014, one of the Nature publications says that there is a semi-synthetic genetic alphabet. Semi-synthetic genetic alphabet means, you see, AGCT, adenine, thymine, cytosine, guanine are natural. If the natural alphabet are chemically synthesized, it is chemically synthesized. You have seen many evidences, minimal genome, all these things. So the alphabet, the chemical nature of adenine is changed. Chemical nature of cytosine, guanine, thymine is changed. Then it is known as unnatural nucleotides. See, a bacteria, a synthetic bacteria, synthetic virus with a natural nucleotide, our body can recognize, because our body immune system can recognize, can responsible, or can consider, can act only on the any pathogen that is made up of adenine, thymine, cytosine, guanine. If a pathogen is not made up of unnatural nucleotides, how our immune system able to recognize it? So what is the level of we are now? You might have seen many news and expectation that now we have the COVID. Another we can have a very fatal infection, maybe made up of a synthetic virus with unnatural nucleotides. How we can tackle it? So that requires strategies. So such a sort of unnatural nucleotides and synthetic genomes, that is the beginning of synthetic life on the Earth. So the aliens means we cannot expect, as Stephen Hawking said, the aliens will come from other planets. No, I would suggest, or I would consider, the aliens will develop from our own Earth by our own inventions. It is not from other planets. So see, CDC made synthetic Ebola virus already, and now the synthetic SARS-CoV-2 is available. Many synthetic viruses are available. The synthetic viruses are originally developed for the discovery of vaccines, but that for good reasons. But the synthetic virus with unnatural nucleotides, what will happen? That is the question mark. You have to think about it, and you can discuss about it. So I told you there is a possibility of zoonotic evolution, means the virus evolved from the bats or the animals to humans. But another chance of evolution is human interventional evolution, means the synthetic virus created in the lab, and the lab-based virus escaped to the earth or purposefully escaped to the earth is again disastrous. So if you want much about it, you can see we had one publication about the zoonotic evolution implication of microbiome in viral transmission infection. And if you are interested in really synthetic biology, some of you just go through the DBT. You just type IBEC. IBEC means Indian Biotechnology Engineering Competition. So you will be 5 students and a guide supervisor. Immediately they will be awarded 10 lakhs for their idea. What is the idea? I told you at the beginning, the synthetic genome is made up of lot of synthetic gene constructs, synthetic genes and pods. Pods means respirators. So the respilator, all are oligonucleotide designs. If you design a oligonucleotide and you can innovate a new oligonucleotide concept, a promoter switches, or a new gene concept, you can apply to the IBEC competition. You will be awarded for 10 lakh rupees immediately. Later, you will be funded further funding. So you are able to develop your synthetic biology research. You can promote synthetic biology research in your team. I wish you all good luck. If you have any questions, please.
Thank you so much, sir, for your nice explanation about the synthetic biology, especially gene engineering and design, Fluxomics, GenoCAD, Tinkercell softwares, and its application in greener bioprocess. And also you explained about how to cheat death and the gene editing in babies and artificial womb cysts and the metagenomics application in various aspects, structural and functional based. And also you conclude with the COVID-19 is a synthetic virus. Now I would like to call participants. If there are any queries, please unmute yourself. You can put your questions. Sir, there is a question in chat box by Kriba Shankar.
Yes, sir.
Does this metabolic pathway going to be same for all microbes for a similar substrate? Also, where to get the metabolic pathway for a specific microbe or product? Please let me know on how to divert electron flux in metabolic pathways.
Okay, so see, your question is like the metabolic pathway or specific. Yes, I told you metabolic pathway engineering for a specific product only. So we have to redirect entire metabolic pathway to a specific product. For example, polyhydroxybutyrate. That was an example I cited, or a synthetic rubber, or synthetic silk, or a hormone, or a enzyme, whatever may be. So what our aim is, if the entire metabolic pathway is directed to a specific product, we will get the maximum production of the product. For a good example, in reality, see why the biofuels, why the biofuels are not in the market, in normal market. The reason is economy. Even the strain which produces ethanol or a alcohol or a hydrogen-based in a biofuel, the strain will die over the accumulation of the same alcohol, maybe 20% limit, it will die. So we create we engineer the pathway, thereby the strain should survive able to 40% in accumulation of its own metabolic end product. So similarly, the entire metabolic pathway is directed towards a particular product. So we will get an economic bioprocess and economic production process. There, that should be a greatest change in the entire industry. So all industry will have a definitely a drastic change if you have a such 1 minute, please. 1 minute, please. Important. Yeah. So see that is the reason. But as I told you, it is very interesting. See, the flux balance is the problem. So once our product accumulated beyond certain limit, whether the bacteria will survive or not. So that require you have to work it on flux balance analysis. There are a lot of opportunities in this field. It is a very new field. You can start working on it.
Thank you, sir. One more question. One more question from chat. Is not it possible for a naturally developing fetus to be prevented by HIV during gestation period via synthetic biology?
That level technology we don't have, but once after the current Great Winter project, we may have some technique. Actually, I want to say one of the technique, gene silencing techniques, that also awarded Nobel Prize, RNA technologies. But RNA technologies are obviously not successful so far. So such technologies are required to treat or silence a gene or introduce a gene even after birth. But after birth, we don't have much gene editing technology. So everything will happen before birth, at the fertilization level itself. Yes.
Thank you, sir. One more question from Dr. Vijay. Is gene editing in germ cell is allowed in India?
India, the gene editing is actually not in front here. Actually, in germ, it is allowed. Actually, gene editing for at the cell level is allowed, but through ethical procedure. So we have a strict ethical procedures are there. What's the purpose of gene editing? So if the gene editing maybe for example, see the COVID time, the many people are working for developing a vaccine. They are working with the germ cell and editing. Definitely, it is allowed.
Yes, thank you, sir. One more question from the same person, Dr. Vijay. Whether the metabolic engineering product are functionally as of natural product or there is no modifications?
Very good question. Actually, in artemisinin, the bacteria will produce artemisinic acid. It may require definitely some chemical modification as the drug. So the title is drug engineering. So definitely we may not get the drug. So we will get the precursor for the drug. So further maybe for example artemisinic acid may be added with some salt to create the real chemical product for market, pharmaceutical products. So it is possible. There is definitely requires some chemical modification in the chemical synthesis level.
Thank you so much, sir. Is there any other queries from participants? Okay, thank you. I would like to express my sincere thanks on behalf of the Dr. NGP College of Arts and Science and the Dean of our college to accept our invitation. Thank you so much, sir.
Thank you, sir. See you all.
Thank you once again. I request all the participants, please join our next session at 2:15 p.m. For the third session. Thank you, one and all. Thank you. Thank you so much, sir.